This is the BioCentury Show. Brought to you by BioCentury Grand Rounds, returning in 2025 to Chicago, Illinois this June and in Cambridge, UK this September. Connect with VCs, academic innovators, and biopharma R&D and BD leaders to explore groundbreaking advancements in early-stage R&D and dive into disease biology, therapeutic modalities, and enabling technologies. Learn more at biocenturygrandrounds.com.
Hello and welcome to the BioCentury Show. I'm Simone Fishburn, Editor-in-Chief, and I am delighted to be joined today by Bahija Jallal, CEO of Immunocore, one of the most prominent biotechs in the UK, and more broadly than that, in the field of TCR immunotherapies. Bahija came to Immunocore in 2019, having previously been president of Medimmune, AstraZeneca's global biologics R&D unit. And of course, she was an EVP of AstraZeneca and member of its senior executive team. Bahija is also on the board at Elevance and a member of the board of trustees at Johns Hopkins. Now, at Immunocore, Bahija has led the creation of the first TCR therapy to be approved, Kimmtrak, which uses the company's soluble TCR receptor, or T-cell receptor technology. Kimmtrak was approved in 2022 for uveal melanoma and is in additional phase 3 trials as Immunocore extends its pipeline to other cancers as well as to infectious disease and autoimmunity. And listeners of the show will know that all of that is going to be relevant to the conversation that we're just about to have. Because there's obviously many other things from your background, Bahija, that I could include. We just don't have time to do all of them. So I really want to start by going a little bit back in time. You have achieved, obviously, a tremendous amount since joining Immunocore. But, you know, when you made this transition, I am sure you had the pick of the crop. And I want you maybe to walk us through what factored into your decision. You went from a big pharma and Medimmune within a big pharma and to a biotech company. What factored into choosing this company, and has it turned out as you expected?
Well, first of all, thank you for having me. It's a real pleasure to be talking to you. Yeah, you know, I always consider myself within a biotech within pharma. So I was in Medimmune, and it was at the time where we achieved mostly what we really set ourselves to achieve. And so I was not really looking for a job, but I had several opportunities. And this one, I have to say, the first time they approached me, I basically said no, absolutely not, because of, you know, because I didn't even have the time or to look at the science. The governance and everything that was happening was not great. And then Sir John Bell, who is the chairman of the company, lured me into a meeting with him in London. And he showed me data. He was actually going to the ASCO, and he said, I'm just going to share this data with you. And what really struck me, this was an, patient showing a liver metastasis that's responding to the drug. Now, as you know, we were just coming out from the checkpoints inhibitors. They've been just approved. And we know, as much as the other companies knew, that when there is a liver metastasis, patients don't respond. So it was something really special that got my attention. After that, I started, talk to investigators and talk to others. And it's really, I knew that this platform is special, that has, and basically I went to the company for the science, definitely.
Well, one of the reasons I asked that, and it's interesting that you answer largely because of the science, but one of the reasons that I asked is that what you have done is taken what is a new modality. So it is a new molecular species with soluble TCRs. And you've taken that to market. Now, we've written at BioCentury about this extensively, including in our back to school last year. there's a lot of issues around that. You have to pick the right target. We talked about that. You have to pick an indication. And I want you maybe to talk us through that decision making, because I know from our earlier conversation that there were steps along the way in this path that what you've sort of indicated kind of went against the grain, that a farmer might not have moved ahead based on the ORR data that you had, but you did. So maybe tell us how it played out, picking those things and then deciding when to stick with it.
Yeah, no, definitely. So, the reason actually, when you looked at just the ORR, even before accepting the job, the ORR was really low, 5%, or, at the best, it was 5 to 8% ORR. But what was really key is talking to the investigators. The investigators, actually, before I even took the job, they were saying, we see this drug, or we have patients, they should not be alive today who are alive, and they feel well. And I think this is really something important. We always have to listen to investigators because they have basically the finger on the pulse. They're talking to the patients. And then coupled with the mechanism, that basically something working in a very cold tumor. So if we talk about the hot tumors like melanoma, cutaneous melanoma is at one side of the spectrum. The uveal melanoma, where we were, is the coldest you can have. And to have an impact there that basically absolutely said there is something special about this drug, about this mechanism that we need to at least explore. And that's exactly, what we did.
Maybe just for some listeners, you want to walk through the sort of hot versus cold tumor and then how this therapy maybe mechanistically turns the hot.
Definitely. So the hot tumor is basically when T cells are there, they're inactivated, and that's the whole mechanism of the checkpoints inhibitors. But at least the immune system has made it to the tumor, can see it, but then is inactivated by the tumor, right?
So in that case, you're just activating something that's there. You're like unleashing the brakes on it with the checkpoints, right?
Absolutely, with the checkpoint. So you have a good starting point, if you will, because it's a hot tumor. The cold tumors, the T cells don't even make it to the tumor. So you are completely in the desert where it's completely cold. And that has been really a challenge for the checkpoints inhibitors. So to see something that has some activity or at least showed activity in phase 1 into uveal melanoma, that was really the intriguing part for me.
So, I think this is a very, very interesting area. I do want to come back to some of the other indications in a minute, but I want to ask you this because you, Immunocore, took this product to market by yourself rather than with a pharma partner. Now, I did an analysis quite recently, and we've seen a growing number of biotechs do this. So, in fact, half of the FDA approvals for NMEs in 2022 to 24 were from biotech sponsors. And maybe you can talk a little bit about somebody who's got this long-standing understanding of the industry. You came from a pharma with AstraZeneca. Was this a decision you made early on? What was behind it? What do you think is in this trend? And talk a little bit from the viewpoint of a biotech CEO doing this.
Yeah, so I think I'll talk about the scientist in me and then the business part of me as well. The scientist in me, really believes that there is a continuum in coming from research all the way to commercial and the information back. I think it's really important to just having a successful phase 3 is not enough. Once you get into the market, you get more information. So for us, it's a new, absolutely we were pioneering this area. Nobody has done it before. We needed to learn more into the market. I think when you look at from the business point of view, it was worth also taking the chance and actually doing it because, again, it's a small indication coming from from pharma, sometimes that gets deprioritized very quickly. So it's not, we're going to care about it a lot more than if we do the big pharma. So there was these two factors that really, and the approval and the support from the board that got us to take that chance. And actually, we don't regret it today.
And your sales are increasing. You've obviously had to build a sales force within that. Was that, something very new for the people on the team? That's a very different kind of company than just an R&D company, right?
Definitely. And I think it's still, I learned a lot in AstraZeneca, definitely, I think because we were interacting with our colleagues in commercial and everything. But it's like when we went about developing this drug, we just were not afraid to take risk. But we brought people who were in big pharma, who've done launches before, and so we're really happy where we are.
So let me go back to something that you alluded to before, which is, I know top of mind for you, which is how data from the clinic should be informing research. For me, this is actually the real definition of translational research, is it starts in the clinic and then you go back and you look at these opportunities and what you're learning from trials. So there's two things I want you to talk about. And then I sort of have a follow-up question, but, there's T-cell fitness, right? How that fits into how you design your next programs and trials. And then after that, we'll talk a little bit about ctDNA, circulating tumor DNA. So tell us just about T-cell fitness.
Yeah, so we really looked at, what's happening in our trials, why we see a different impact, or we see a different way of looking at efficacy, if you will. So we call it the disease control rate in general. So if we take patients that are they don't have the 40% reduction where you actually say that you have the PFS. You basically look at patients that hover around the 30%, and they actually benefit. We have the phase 3 to prove it. We've done all that. So we looked at, we always say that immunotherapy works better as you get in first line and things like that. And so there we really just looked at the T-cell signature and what's really important. And what we see, actually, we reduce it to what we call our T-cell fitness, that basically T cells that are less differentiated, that can, have a stem cell-like fitness, that basically are at the beginning, at the first line and earlier lines. The patients who have that, I would say it as have more strong immune system, work better. So I think it makes sense, but we had to do the experiment to prove it.
So one direction I want to go with this is, one of the things that we hear from smaller biotechs is that, on the one hand, there's this idea that during clinical development, you want to be gathering a lot of data so that you can do this kind of research. Right. But sometimes we hear biotechs telling us, that is the last thing I'm going to do. I'm not going to get any single piece of data that FDA doesn't absolutely have to see for this, because they worry that they might create a signal that then needs exploring. How do you think about that? And, do you think that that is sort of appropriate for some companies or misguided, or what are the risks?
Well, I really don't see a risk in producing data. I think we owe it to ourselves, we owe it to the patients and everything, to understand what we're doing, right? And I think when we have the opportunity, absolutely, that becomes really important. Otherwise, it's a binary. It works, it doesn't work. And then you don't really advance the field. And that's the problem. So for me, instead of doing 5 trials, I'll do 2, but really well informed, I think, right. And following the science is the most important thing.
Okay, well, we're going to go to a break in a minute, but before we do that, I do want to talk about ctDNA, circulating tumor DNA, because that is a biomarker in the KIMMTRAK trials, as I understand.
Right.
And I sort of want you to talk about that, but also, like, the pace of progress with that as a biomarker. How do you think about that? It seems that there's been promise of circulating tumor DNA for a long while, but I'm not really sure where we are as a field in terms of that coming to fruition. And how do you envision it being implemented in the next, let's say, 3 to 5 years?
Yeah, so I think ctDNA, as I said, there's a lot more progress done, but to get it to the final point, which basically you take it as a surrogate marker for efficacy, you need a lot more, you need a lot more prospective studies, if you will. So there was a lot more done with the ctDNA in lung. With the Friend of Cancer Research and really pulling the data from different companies and showed basically that, that's, I think, the largest data set that exists there. And I think the problem is always when you do these exploratory, biomarkers, you have to then do a prospective study to show that it's a long haul, if you will, to get to the point where it becomes a surrogate marker. But for us, it's a really good guidance, and we will pursue it until we generate the data to show that there is a different way to actually then resist to look at the effect of some of these drugs like ours, and especially platforms like ours.
So, what do you anticipate? Do you think it's going to become a lot more common in the field? Do you think that it's sort of going to reach a tipping point?
I think so. I really believe so, because, especially as we get earlier and earlier into the adjuvant setting, that becomes a really great tool. And, because as we know, cancer, if we get it early, we have more chance to have an impact. And that's really what we should all be aiming for, getting ourselves out of business by going into prevention,
Absolutely. So we're going to take a brief pause here. And when we come back, we're going to talk about some of the other therapeutic areas of Immunocore.
Grand Rounds. BioCentury's R&D conference will return in 2025 to the US and Europe to debate key bottlenecks in translation and unlock scientific breakthroughs with commercial potential. Connect with VCs, academic innovators, and biopharma R&D and BD leaders to explore cutting-edge advancements in early-stage R&D as we dive into disease biology, therapeutic modalities, and enabling technologies. Join us in Chicago, Illinois this June for the second BioCentury Grand Rounds US, and in Cambridge, UK this September for the inaugural Grand Rounds Europe. Register and learn more at biocenturygrandrounds.com.
We are back at the BioCentury Show with Bahija Jallal, the CEO of Immunocore. And we've talked about the oncology programs, but, I think maybe people know a little bit less, in fact, that you have programs in two other major disease areas, infectious disease and immunology. And I want to start with the infectious disease pipeline, which I think is very exciting. And you have new data in HIV. Now, I think it's interesting because many people think that HIV is a problem that's really solved. But obviously, you are going after a functional cure. I think the aim is to reduce or eliminate sort of rebound competent reservoirs. So, Bahija, and I know people have been trying to do this for a while. Are you banging your head against a brick wall, or what does your program do differently?
Yeah, I think it's just maybe a step back on what we're trying to achieve and what has been achieved in HIV. I absolutely, applaud the fact that, the HIV is no longer a death sentence, right? So it's a chronic disease. However, we have the antiretroviral work perfectly. But the problem is the minute you stop, the virus comes back. And the issue is that the virus hides in what we call the reservoir, so CD4 cells and things like that. And it says it's dormant, so it's a very low copies, if you will. And the issue is the compliance when you are outside of, the U.S. or other parts of the country. You have also, there is an infection or inflammation that's also this long term. You have to take the medicine all your life. So why did we believe that we could maybe at least do the experiments? Is the fact that these one thing that's really important about our platform is it's very, very potent. We get to the picomolar level. That means we need 5 copies per cell to kill the cell, right? So this is how we do with the TCR. We have the TCR binding to the cell and we bring the CD3. So because we are so potent, we thought if we go after the reservoir, not the virus itself, but after the reservoir and eliminate the reservoir, we have the chance to bring about a functional cure. So we set up to do the experiments. We had real enthusiasm, and that's really important, real enthusiasm in the first single ascending dose. We did it very quickly, and then we did the multiple ascending dose. And the way we did it is to have basically the first 12 weeks, we have the antiretroviral plus our compound to go after to ask the question, can we modify or eliminate the reservoir? Nobody has done it reliably so far. And then we stop everything. And then we say, okay, how fast can we delay that rebound or not? And there was a meta-analysis that showed that if you stop just from the retrovirus, after 2 weeks, you get, patients completely coming back, and by 8 weeks is 90%. So the first preliminary data we went from 0 to 300 microgram. We're talking microgram. This is how potent this drug is. We did the multiple ascending dose, and we did up to 300 microgram, and it was safe and also showed biological activity. And now we're going even higher, so which really speaks volume about the safety. What we've shown is we actually can reduce the reservoir. We also show that we have delay of the virus rebound. We put a limit at 12 weeks, and we had some patient, 1 patient or 2, 3 patients actually, that those dependent delay of the rebound, which is really unprecedented. It's great. That's the first time. But this is just the beginning. So we are going back into increasing, escalating the dose, and hopefully, we presented this at CROI, and there was really positive reception. So we're happy about that.
Yeah, well, I do think it's a very interesting program. We could probably talk about that and other programs from a while, but I want to jump from here to autoimmune disease. Now, this is a huge area right now.
Yes.
And I'm going to start actually by a kind of high-level view on this area, because it's really been mostly for a long time the TNFs and then cytokine modulators after that have dominated this field, and patients quite often cycle through those. And so they're very large population diseases. There's not much precision medicine in that area. But tell me how you think that treatment paradigms might be changing in autoimmune disease.
Yeah, it's really biologics, I think, made a huge difference.
Right.
If you think about Humira and others, that's we're starting to see and think about, disease modifying, agent. And I think the biologics really have that. I think one of the issues, especially at the beginning, was it's not so much. The efficacy definitely is there, but the safety also for the small molecules and other agents in autoimmune diseases, because these are chronic diseases. You take them for a while, and
they're very blunt tools, right?
Completely blunt tools, absolutely. Right. Like the slash hammer, I call it. The steroids. You want to bring that inflammation, but then you're also bringing it in the whole body. And I think the biologics are starting to transform that. But I think this is an area, in my opinion, that also can benefit a lot more from, precision medicine, as you said, and actually selecting the patients, because they're not always the same. A lupus patient is not the same as any other lupus patient. So the advancement that we have in cancer, I think absolutely autoimmune will benefit from that as well.
Well, it's funny that you talked about the patients being so different, because I'm going to ask you about your programs, because you've got one in type 1 diabetes and one in atopic dermatitis. And those are very different target populations and very different biologies. So talk about that decision making. How did you choose those indications? And how much does the learning from one program kind of inform the other, or are they just completely separate?
Yeah, I think the overarching principle is to look at organ-specific down modulation of the immune system, right? Down modulation of the immune system. That's what we're trying to do. So in that respect, both of them are in the same boat, right? And again, to avoid that slash hammer that you get. So it's more. So this is our foray into more precision of precision medicine into autoimmune. So we took type 1 diabetes because, there we have a peptide that's presented very highly and only in beta cells, basically in the pancreas. So there we have also a biomarker, right, the C peptide. So that means in autoimmune, it's really important that you have a good translational to see if there is higher probability of success to get to the next phase, because these are much larger trials and so on. So that for type 1 diabetes, both have the same effector, which is the PD-1 agonist. So we are trying to do the opposite of cancer, basically, is activate, bring down the activity of the T cells.
Activate the brakes to
Exactly.
Stop. Right.
So that's what we're trying to do. So that's type 1 diabetes, and we have all that. And I think we can know, or we will know, early on if this is working or not because of that C peptide. And then we had also through, all these methodologies that we have in the company of looking at peptides that are specific, organ specific. So we have one for it's the CD1 basically in the skin. And there, CD1 has been targeted before, but we believe that there are two. We completely want to block the whole pathway, the two pathways in the CD1. And again, we'll go to skin diseases because they're T cell driven. So that's one of the common things as well. They're T cell driven, and it is possible to have biopsies and again do more of the translational early on to find out if it's working.
Yeah, I mean, I think we're continuing to watch that, the whole area of, B cell reset and, various other novel ways of addressing autoimmunity. But, we have just a couple minutes left. And to end, I want to take a step back and talk to you about being a biotech CEO today. It's an extended bear market. Many people thought several times that we were coming out of it. We talk about it as Lucy in the football. We've kept thinking that the markets would, get better, and they keep getting hit again. Immunocore might be in a slightly different position, but generally we are in a highly uncertain time with respect to FDA and the future of science and NIH. Now, I understand Immunocore is based in the UK, but much of it is centrally relevant. So I just want to ask you, as a leader managing through this, what are you hearing from other CEOs? And what are you hearing from employees and colleagues? What is the mood, and how do you manage a company? You're doing fine, but not everything around you is that great. So how are you managing through this?
Yeah, no, absolutely. I think, one is, I am so glad we made the decisions that we made when we made them, which is, commercialize our drug, doing a convert last year just before the market went down. So my philosophy has been, you go get money not when you need it, but you get money for these kind of times, right? The second is, I think people are really resilient. You cannot be in this profession if you're not resilient. Having said that, I think there's just a lot of moving parts right now. What's going to happen with tariffs, what's going to happen with the NIH, we live in an ecosystem and we're interdependent on the universities, on the NIH, and that's why, and Oxford University and all that. And so that system has to work and has to be, a strong ecosystem. When it's not a sure thing, then you are really walking into a new territory. So what we do, obviously this is on the mind of everybody and the employees, but what we do is this is where we double up on bringing the patients and reminding people why we're here every day. And it doesn't matter how our anxieties can be high or not, but people need these medicines, and that's why we're here. We're here for the patients.
Well, I really can't think of a better way to end this conversation. I hope we'll have many more. We're very interested in watching to see how Immunocore takes these things forward. And thank you very much for joining us.
Thank you so much, Simone. I really appreciate it.
Thank you.
Thank you.
Brought to you by BioCentury Grand Rounds, returning in 2025 to Chicago, Illinois this June and in Cambridge, UK this September. Connect with VCs, academic innovators, and biopharma R&D and BD leaders to explore groundbreaking advancements in early-stage R&D and dive into disease biology, therapeutic modalities, and enabling technologies. Learn more at biocenturygrandrounds.com.